Golgi apparatus is an organelle in eukaryotic cells that consists of 5 to 8 cupped or flattened membranous sacs called cisternae. Lumen are the open spaces within these cisternae. Golgi apparatus has two distinct poles, the cis face and the trans face. The chemical makeup of both cis and trans faces are different, as are enzymes within the lumens. The function of the Golgi apparatus is to modify, store, and route products of the endoplasmic reticulum. Vesicles containing proteins and phospholipids and other products break free from the endoplasmic reticulum as they travel to the cis face of the Golgi body. Within the Golgi apparatus, enzymes add to the molecules or chop pieces off them to get them to the proper shape and size. Enzymes also subtract or add sugar monomers to carbohydrates or glycoproteins. The final products leave the Golgi apparatus through the trans phase via vesicles. The Golgi apparatus also adds tags which direct the vesicles, such as phosphate group. The Golgi apparatus is located in between the endoplasmic reticulum and the cell membrane. This is because it receives vesicles containing molecules that need to be processed from the endoplasmic reticulum. The final destination for many of the molecules processed in the Golgi apparatus is the plasma membrane, therefore it is located near it. The Golgi apparatus is critical in the sorting, modifying, and directing of fats and lipids, which it receives through vesicles. It aids in the maturation of lysosomes and gets molecules ready to travel outside the cell to carry out various functions. The Golgi apparatus was discovered by Camillo Golgi in 1898.
Lysosomes are the key digestive organelles in animal cells. Their structure is very simple. The outside consists of a phospholipid bilayer, and the inside consists of a mixture of hydrolytic enzymes. This mixture is also very acidic. That is due to the fact that these enzymes digest the best under acidic conditions. Also, the enzymes have 3D shapes in order to keep the weak bonds away from the low pH of the lysosomes inside, which prevents denaturation. The function of the lysosome is to digest macromolecules in animal cells. First, macromolecules are engulfed by the cell by the process called phagocytosis. The food vacuole is then fused with the lysosome, which releases its hydrolytic enzymes into the macromolecule. These hydrolytic enzymes are essentially enzymes that speed up hydrolysis, decomposing the macromolecules into amino acids, simple sugars, and other monomers that the cell can use. Lysosomes also help with the process of autophagy. This is when old cell organelles and parts that can no longer be used are engulfed in vacuoles, which the lysosome then fuses with. The organelles are decomposed into their simple parts that are then released into the cytosol for further use by the cell. As stated earlier, the lysosome depends on the food vacuoles to carry out its processes of digestion. Also, the lysosome depends on the Rough-ER and Golgi apparatus to come into existence. The Rough-ER creates the lysosomal membrane and the hydrolytic enzymes, which are then sent to the Golgi apparatus for construction. The final product is the lysosome, and as can be seen, the lysosome is located in the cytosol. Lysosomes are obviously important to conduct the digestion in animal cells. Also, all the lysosomes in a cell can simultaneously release their contents in order to autonomously destroy the entire cell if the cell has become worn out, harmful, or useless. Also, if hydrolytic enzymes are missing or inactive, then the substrates will not be digested. This interferes with the cell's function, and diseases can be caused such as Tay-Sachs, which is found in isolated communities such as the Amish or the Cajuns. Also, an interesting fact about lysosomes is that their name comes from two Greek words, lysis meaning destruction and soma meaning body. Literally, they are destructive bodies.
The vacuoles are made of cell membrane and store food, water, and other materials. They are a coalescence of smaller vacuoles, or vesicles. The main functions of vacuoles include containing waste, containing water in plants, maintaining the turgor, or the pressure of the cell contents against the cell wall in plants, containing small molecules, and exporting unwanted substances from the cell. In amoeba, food vacuoles engulf food particles through endocytosis. Then lysosomes attach to the food vacuoles and digest the food inside the vacuole. From there, it is eliminated through the cell by exocytosis. Vacuoles also contain pigments and give many plants their color. Plant cells have one large central vacuole that can occupy up to 80% of the cell's volume. The tonoplast is the cytoplasmic membrane surrounding a vacuole, separating its contents from the cell's cytosol. The central vacuoles regulate pH as well as the rigidness of the cell. As you can see, the central vacuole of a plant cell pushes all of the other organelles up against the cell wall. As water diffuses into the vacuole, it becomes larger. Vacuoles are located prominently in the center of plant cells. They interact mainly with molecules that need to be regulated within the cell. They are made of smaller vesicles that are formed in the Golgi apparatus and the endoplasmic reticulum. Vacuoles are important in maintaining the overall rigidity of many plants, storing food, and excreting waste products. One interesting fact is that the name comes from a Greek word meaning tension, because they provide tension and rigidity within plant cells.
The Golgi apparatus, lysosomes, and food vacuoles are greatly interrelated organelles of the cell. First of all, lysosomes are vesicles constructed and formed by the Golgi apparatus. Then, these lysosomes fuse with the vacuoles in order to carry out digestion of food absorbed by the cell.